A-level biology, cell structure. You need to know the structure and function of each of the organelles in a eukaryotic cell, which includes plant cells, animal cells, algal cells, and fungal cells. Let's start with the cell surface membrane or plasma membrane, which controls the movement of substances into and out of cells. It's made up of a phospholipid bilayer, with phosphate heads which are polar and hydrophilic on the outside, and fatty acid tails which are nonpolar and hydrophobic on the inside. Within the bilayer, there are proteins such as structural proteins, carrier proteins, and channel proteins. There are also carbohydrates, glycoproteins, and glycolipids, which function as receptors, and cholesterol, which helps to increase the fluidity of the phospholipid bilayer. The nucleus controls the activities of the cell by controlling the transcription of DNA. On the outside, there's the nuclear envelope, which is a double membrane containing nuclear pores allowing substances to enter and exit the nucleus. The chromosomes are made of protein-bound linear DNA, and these proteins are called histones. The nucleolus, the site of ribosome production in the cell, can be seen in an electron microscope image as the darker region within the nucleus. The mitochondria is the site of aerobic respiration in the cell to produce ATP. They are oval-shaped organelles bound by a double membrane, with the inner membrane folded to form cristae with the matrix on the inside. The matrix contains mitochondrial DNA, separate from the DNA in the nucleus, which encodes the enzymes needed for respiration. Chloroplasts are found in plant and algal cells, and they are the site of photosynthesis. Like mitochondria, they are oval-shaped organelles, bound by a double membrane. They contain thylakoids, flattened membrane sacs containing pigments such as chlorophyll, which is stacked to form grana. The chloroplasts are filled with fluid called stroma, which contains the enzymes needed for photosynthesis. The Golgi apparatus processes and packages proteins and lipids, and also produces lysosomes. It is made of fluid-filled flattened sacs and has vesicles surrounding it, which transports the products it produces. Lysosomes digest worn-out organelles and pathogens, and have a single membrane around the outside. They contain hydrolytic enzymes called lysozymes to help them digest material. Ribosomes are the site of protein synthesis. Eukaryotic cells contain large 80S ribosomes compared to smaller 70S ribosomes found in prokaryotic cells. Ribosomes are made up of two subunits and consist of protein and RNA. There are two types of endoplasmic reticulum, the rough ER and the smooth. The rough ER folds and processes proteins, whereas the smooth endoplasmic reticulum processes and packages proteins and lipids. Both organelles are made of membrane-bound, flattened sacs. The main difference is that the rough endoplasmic reticulum has ribosomes, whereas the smooth ER does not. The cell wall, found in plants, algal, and fungal cells, provides structural support. In plants, they are made of cellulose molecules joined by cross-linkages to form microfibrils, whereas in fungi, they are made of chitin. The role of the vacuole is to keep the cell turgid. It has a single membrane on the outside, called the tonoplast, and is fluid-filled and stores nutrients such as salts, sugars, and amino acids. In complex multicellular organisms, eukaryotic cells become specialized for specific functions. Specialized cells are organized into tissues, which are organized into organs, which are organized into organ systems.